Hello students, welcome to InstaOwl Science class where we believe in democratizing education for all. Students, we know the small changes that happen during the reproduction are called variations, and we have seen how they help organisms adapt and survive when conditions change. Now, let's see how some of the simplest organisms like amoeba and yeast reproduce and what methods they use to create more of their kind. Students, let's notice bubbles in rising dough or fuzzy spots on old bread. They might seem like signs of something going stale, but actually, they are proof that tiny living organisms are at work, doing something essential, reproducing. We'll now discover how single-celled organisms like yeast, fungi, and protozoa use different methods to create more of their kind. And to understand this better, we'll start with two quick and simple activities. First up, in this activity, we need to take about 10 grams of sugar and dissolve it in 100 ml of water. Then, pour 20 ml of this solution into a test tube and add just a pinch of yeast granules. Seal the mouth with a cotton plug and place the tube somewhere warm, like near a window or a lamp. After about 1 or 2 hours, take a small drop from the tube, place it on a slide and cover it with a cover slip. Now, here's the exciting part. When you observe it under a microscope, you'll spot something amazing. Tiny yeast cells forming small buds right off their surface. This is budding, a common method of asexual reproduction in yeast. Now, you must be wondering what is asexual mode of reproduction. Let's understand the basic definition of it. Asexual reproduction is a mode of reproduction in which a new offspring is produced by a single parent. The new individuals produced are genetically and physically identical to each other. That is, they are the clones of their parents. Asexual reproduction can be observed in both multicellular and unicellular organisms. So, budding is also an asexual mode of reproduction where a new organism develops from an outgrowth or bud on the parent organism. We will study this ahead in detail. Next up, for the fungus activity, we need to take a slice of bread, dampen it slightly with water, and place it in a cool, moist, and dark place, maybe inside a closed container or a cupboard corner. Just observe it daily with a magnifying glass and record what you see. After a few days, usually by the third or fourth day, you'll notice fuzzy green or black spots growing on the bread. That's fungus, and it reproduces by spores. This happens because in single-cell organisms, reproduction happens differently from humans or animals. The most common way is through fission, which is simply the splitting of one cell into two or more new cells. Let's take another example of bacteria or protozoa. They don't need any special setup to reproduce. They just grow, then split straight into two equal halves, like slicing an apple into two pieces. This is known as binary fission. Now, let's look at this activity to understand binary fission in depth. First, take a look at a permanent slide of amoeba under the microscope. What do you notice? You'll see a single irregular shaped cell with a clear nucleus. That's how an amoeba usually looks. Now, observe another slide, this time of amoeba during binary fission. Look closely. You'll see the nucleus dividing, followed by the cell pinching inward and eventually splitting into two daughter cells. And that's your result. Amoeba reproduces by binary fission, where one cell divides into two identical cells. It's simple, silent, and happening right before your eyes. The cell can divide in any direction. There's no fixed angle. It simply splits into two wherever it grows best. And both parts then continue life as independent organisms. But not all unicellular organisms are this random. Some, like Leishmania, are more organized. Leishmania parasites are the cause of Kala Azar, which is a serious and potentially fatal disease that affects internal organs like the spleen and liver. They have a special whip-like tail called a flagellum at one end. Because of this structure, it doesn't divide just anywhere. Their division follows a specific direction aligned with how their body is built. So, even though the method is still binary fission, the pattern depends on the organism's structure. Now, let's take a quick turn to Plasmodium, the parasite responsible for malaria. It doesn't stop at 2. Instead, it divides into many daughter cells at the same time. It's like dozens of copies forming all at once inside one cell. This process is known as multiple fission. Before we wrap up, there's one more method worth noting: budding, seen in yeast. Instead of splitting in half, yeast grows a small bud on its surface. This bud slowly grows, detaches, and becomes a new individual. We'll take a closer look at this unique method in the upcoming video. Now, let's shift our focus to the instalites. We first saw asexual reproduction in single-celled organisms where new individuals are formed without any involvement of another cell. Then we explored binary and multiple fission in unicellular organisms, where cells either split into 2 or many depending on the species. And finally, we looked at budding in yeast, where a small outgrowth forms and grows into a new cell. Keep growing with InstaOwl. Bye!